Hey y'all, my name's Cole Crenshaw. I'm a biochem major here at KU, and I've been working in Dr. Choi's lab this year. I kind of jumped right into the middle of a research project, but I want to talk about our research question real quick, and that involves telomeres, which are repeating regions at the end of eukaryotic chromosomes that protect the DNA from degradation upon replication. So each time you replicate your cell, you also have to replicate your DNA, and over time in eukaryotes, this wears down the ends, and this would be bad, especially if you have genes there and you would lose those genes and eventually lose functioning proteins, and then the organism would not be able to survive. So, this is rectified by having telomere regions on the ends, which are just repeating regions that can be worn down without having any negative impact. But in cells that replicate a lot or that lead to gamete production, that you're going to pass down that information to the next generation, you want to rebuild those telomeres. And this is done through the telomerase enzyme, which is just a little factory that moves along and rebuilds it with two parts. It has the telomerase RNA, which is just a template, and then it has the telomerase reverse transcriptase, which is a protein that uses that template RNA to rebuild the telomere. And this is where our research question comes from, because the telomerase RNA, that part of the telomere rebuilding enzyme, the telomerase, has called the TR gene. And this gene is typically evolutionarily conserved, which means it doesn't change a lot because it's very vital to survival of the species. But here in this Mimulus genus, we see, especially in between these different species that I'm working with, Lewisii and Verbenaceus, we have variation. Lewisii has a duplication in its TR gene, which impacts the telomere DNA sequence because it uses that as a template. And my research works with the hybrid offspring of these two species. So we took a Verbenaceus and a Lewisii, have their offspring, and back cross that with the Verbenaceus plant. So we're doing this over and over again to get successive generations to eventually produce a plant that has mostly Verbenaceous DNA. That's the normal non-duplicated TR parent. And then we have the few genes, including that duplicated TR from the Lewisii. And we're trying to see if this duplication in the TR has any impact on the growth rate or the growth of the plant. And then we also have a few plants as a control that we're allowing to continue through the experiment without the TR gene to make sure that we don't have any confounding genes or any other variables in there we don't know about from the Lewisii that are impacting results. So I jumped right into the middle of the research cycle and I started conducting growth experiments. So we collected seeds from the previous experiment, we germinated those, watered them, transplanted them, and then took measurements of 100, 200 or so plants over an 8-week period, and then we froze them for analysis. And then this semester, I've been extracting the DNA, running PCR, if that's just amplifying the genes, or the gene we're looking at, the fragment of the gene we're looking at, and then isolating that, and then we are running gels. So this is just to test for the presence of the gene. And you can see that we have a picture in the bottom left of one of the plants we're working with, and then on the bottom right, we have a gel image there. And we have on the top of that, we have the V, or the Verbenaceous TR gene. If they're there, it's positive. If it's blank, it's negative. And then on the bottom, we have the TR gene from the Lewisii, and if that's there, it's positive. If it's not, it's negative. And that just shows us if it's there or not. We can correlate that with the growth data and see if there's any correlation that might lead us to think about causation. And then over the course of the semester, I've learned that research can be hard sometimes. It can be tedious sometimes, but it can be very rewarding if you learn something from it, either in your own personal, like how to research, what it means to research, why it's important, or even just like broadly, research is like the basis of all our textbook knowledge, all of our scientific knowledge. So it's very important. So thank you.